In this video of general biology, we would be discussing cell division. So biology is the only branch of science in which multiplication is the same as division. Okay? When we talk about multiplication, we are talking about cell reproduction. Okay? Cell reproduction occurs when a cell reproduces by dividing into two. Okay? So each descendant cell actually receives a full set of chromosomes and new nuclei, and the cytoplasm divides by a separate mechanism. Okay? So there are actually two methods of nuclear division for cells. So it could be your mitosis or meiosis. Okay? So mitosis is a nuclear division process that maintains the chromosome by doing asexual reproduction. Okay? So meiosis, on the other hand, is a nuclear division process that halves the chromosome number, and this is the basis of sexual reproduction in multicelled eukaryotes. Okay? So it actually precedes the formation of your gametes or the sexual spores, okay, or the sexual cells. Okay? Now let's go to the life of a cell, or let's say this is the cell cycle, okay? So it starts when a new cell forms and ends when the cell reproduces, okay? On the animation on your right, this is actually a cell undergoing mitosis, okay? So first part, or before we go to the cell cycle, remember that your cell cycle would be a series of events from the time a cell forms until its cytoplasm divides. Okay, so as you could see, you have red structure, so that is your chromosomes, and then you would now see the separation. Now you have two cells. Okay, so the green structures are actually your cell. Okay. So we now have three phases for your cell cycle. We have the interphase, mitosis, and then we have the cytoplasmic division or your cytokinesis. Okay? So for the interphase, it is actually the interval between mitotic divisions, okay, when a cell grows. So in interphase, you have three stages. You have the G1 or the first interval or gap phase. So it actually refers to the growth of the cell before the DNA replication. Okay. So next we have the S phase or the interval of synthesis, or basically this is where DNA replication happens, okay? Or S actually stands for synthesis. Okay, yung G for G1, it could actually refer to gap 1 phase. Okay, so for G2, that's a gap 2 phase or the second interval phase, and this is where the cell is ready prepared for division. Okay, now if there are no controls for cell division, there would be a resulting cancer. Okay, so different types of cells actually proceed through the cell cycle at different rates, and if control is lost, okay, if there's an abnormality in the division or there's an abnormality in the cells and these cells continue to divide, then there is now the formation of cancer. Okay, so gene expression controls actually help in regulating the cell cycle at different points. Okay. Now, the chromosomes during the cell cycle are actually pairing up. So the chromosomes of each pair are homologous, okay, or meaning they're the same, except for the male sex chromosomes, okay. So on the picture, you are actually seeing a karyotype. So a karyotype is an image of your chromosomes from chromosome 1 to 23. Okay, chromosome 23 is your sex chromosome. So for the females, it's XX, while for the males, it's XY. Okay? So when we say homologous, these are two members of a pair of chromosomes with the same length, same shape, and same copies of genes. Okay? So mitosis actually maintains the chromosome number and each chromosome is duplicated. Now, each new cell or each daughter cell would now receive identical chromosomes. Okay? So here we have an unduplicated pair of chromosomes in a cell in G1. Then by G2, each chromosome has been duplicated and now it's ready for the cell division. Okay? So after mitosis and cytoplasmic division, each cell would now have a copy of each chromosome into each of the new cells, okay, or each new nucleus or nuclei, I mean. Okay, now let's go to the different stages of mitosis. Okay, So at the end of interphase, a cell's duplicated chromosomes condense tightly in preparation for a nuclear division. Okay, so seen in illustrations, you would see the chromosomes represented as sister chromatids, okay, or two sister chromatids. Katulad nung kanina, you have two lines or two structures, okay? So again, there are 4 stages for mitosis. You could remember this as PMAT. Some would use a mnemonic like P in the math, but simply PMAT for PMAT. Okay, so P for prophase, M for metaphase, A for anaphase, and T for telophase. So now let's go to the first stage, which is prophase. Okay, in some references or in some books, there are actually 5 stages of mitosis, wherein prophase is divided into two. You have the prophase and the prometaphase. But for simplicity, we are just using prophase as one stage, okay, or one phase. Okay, so in prophase, the chromosomes condense, so they are become tightly packed and spindle forms. Okay, so at this point, the nuclear envelope breaks up and the spindle microtubules attach to the chromosomes. Okay, so the chromosomes here become visible as distinct structures as they condense further, as you could see. So in this picture, so on your right, you have the animal cell, and on the left, you have the plant cells. Okay. So the DNA actually begins to appear, or it is like grainy structures at the start, but after or at the start of the prophase, you would see now small lines. Okay. So distinct na yung characteristic or distinct na yung ating chromosome or DNA. Okay. So next we have the metaphase. Okay, at the metaphase, this is the stage where duplicated homologous chromosomes line up at the spindle equator. Okay, so or basically it's just halfway between the spindle poles. Okay, so there's really no metaphase plate, but it's just a reference. Okay, so the chromosomes now line up at the center, and the sister chromatids begin to move apart toward the toward the spindle poles. Okay. So at the metaphase, you would see now the aligned structures of your chromosomes, or they are aligned midway. Okay. So the black structures here are your metaphase. I mean, are your chromosomes lining up at the metaphase state. Here at your plant cells, so ayan, yung red, nasa gitna na din sila. Okay. So next we have anaphase. Okay, so at anaphase, the microtubules separate the sister chromatids of each chromosome and they pull them toward opposite spindle poles. Okay, so the pulling is actually caused by the shortening. Okay, the shortening of the microtubules, and each DNA molecule now is a separate chromosome. Okay, so you would now see the distinction or the separation already. So as the spindle microtubules drag the chromatids toward the spindle poles. Okay, so the spindle poles are at the other side of the cell. Okay, now this is actually a proof that there's no forever. Very sad, no? Naghihiwalay din. May naghihiwalay pa rin pala sa cells. Okay, so just something to remember. So, sa anaphase, walang forever. Okay. So next we have telophase. Okay, telophase is the stage where two clusters of chromosomes now arrive at the spindle poles and they start to decondense. Okay, and now new nuclei are formed. Okay, so what happens is at telophase, if you're going to what do you call this, recall your stages, para lang itong babalik ka lang dun sa prophase, pero baliktad yung nangyayari. It's the opposite. Okay? So at the end of mitosis, the nuclear envelopes form around two clusters of chromosomes, forming two new nuclei with a parental chromosome number. Okay? So for um, animal cells, you would see now this cleavage furrow. So magkakaroon mo na ng cleavage formation before the entire separation. But for the plants, you would now have the formation of a cell plate at the middle. Okay? So here, this animation would show you the entire stages of your mitosis. Okay, so first, let's start. Wait natin. So that is, mabilis lang yung separation, no? So you now have metaphase, anaphase, now telophase, and cytokinesis. So there you have your two new daughter cells. Okay? So cytoplasmic division in plants, as mentioned a while ago, you have now the cell plate forming. Okay? In plant cells, a cell plate forms midway between the spindle poles and partitions the cytoplasm when it connects to the parent cell wall. Okay? So that is now how um, cytoplasmic division is different in your plants and among your animal cells. Okay? Now, sexual reproduction and meiosis. So as mentioned initially, meiosis is responsible for the production of your gametes. Okay, so the offspring of sexual reproduction actually vary in shared traits. Okay, so remember that sexual reproduction is the reproductive mode by which offspring arise from two parents and inherit genes from both parents. Okay, now I would have a short introduction about alleles, but we'll have further discussion of these when you go to our genetics. Okay. So an allele is a form of a gene that would encode slightly different versions of the gene's product. Okay, so alleles are the basis of our traits. Okay, so as mentioned a while ago, you have seen this already, your karyotype. So your paired genes on homologous chromosomes often vary slightly in DNA sequences, as shown by these alleles. Okay, so variation in traits happen because of the new combination of alleles and traits. Okay. So this variation in allele combinations are introduced during meiosis. Okay. So as shown in this picture, which I downloaded from the internet, let's say these are the parents. So you would see now the combination of traits passed on to their child. Okay? So, possibly si daddy ay curly hair na napass niya kay daughter. Then, the skin color of the mother is passed on to the daughter. Okay? Ano pa yung similar traits that you could observe? Okay? So, as you can see, you could actually check your parents and your appearance. Okay? Who do you look like? Do you look like more your father or your mother? Or you are a combination of both your parents' DNA? Okay, so that's one good point to check. Okay. So meiosis, again, as mentioned, is a process that halves or divides the chromosome number. Okay. So meiosis occurs on immature reproductive cells, or also called as your germ cells. Okay. So they form now the male and female haploid gametes. Okay. So their gametes are mature haploid reproductive cell. Okay. So when we say haploid, it's half. Okay. Pero yun naman tandaan na haploid. It's not haploid, but it's half with a P, haploid. Okay. So when you say haploid, it has one of each type of chromosome characteristic of the species. Okay. So your meiosis could also be the spermatogenesis or the oogenesis for the sperm and the egg cells respectively. Okay. Now, meiosis sorts the chromosomes into new nuclei twice. Okay, so there are two divisions for your meiosis. We have meiosis 1 and meiosis 2. And the duplicated chromosomes of a diploid nucleus are distributed into 4 haploid nuclei. Okay, so in the diagram presented, you would now see each chromosome in the cell pairs with its homologous partner. And then, at the first division of your meiosis, they would separate. Okay, and then afterwards, one chromosome, which is already duplicated, would now be further divided into two chromosomes. Okay, so now let's go to the different stages of your meiosis. Okay, so there are actually two nuclear divisions as mentioned. So we have meiosis 1, or also known as the heterotypic division, and it is also called as the reductional division. Okay, reductional division because it reduces the number of chromosomes, or it divides the chromosome. Okay, so in meiosis 2, it is the homotypic division, or also known as equational cell division, because basically at this point you would now just be sharing or dividing the cell into two equal parts. Okay, or each two daughter cell would now eventually produce two more daughter cells. I hope I'm making sense because I don't have a diagram here, but later on you'll see in the next slides, okay? So meiosis actually shuffles parental combinations of alleles, and this would be the reason for the variation in the offspring, okay? So this happens because of the crossing over in prophase 1 and because of the random assortment in metaphase 1, okay? So these are the stages of meiosis 1. So basically, you have PMAT, but it's twice. Okay, so you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then afterwards you'll have prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, so basically it's the same process. However, as mentioned a while ago, in meiosis, there is the combination of alleles. Okay, so there is now variation in our offspring. Okay, so in prophase 1, as you would see, same lang din ang nangyari sa prophase 1 of your, prophase of mitosis. Okay? So the nuclear envelope breaks up and there's also the formation of the spindle poles and your chromosomes are actually condensed. However, one key feature of your meiosis or your prophase 1 is the crossing over. Okay? Now, in this crossing over, you would see now the transfer of genes from one to another. Okay? So, at metaphase 1, you would now see there's a variation already. Okay? It's, for example, the purple chromosome here. This one, you have now a blue chromosome with the purple one. Okay? And vice versa. Okay? And at anaphase 1, it's already assorted. Okay? So, you see here na it's not paired na blue and blue, but rather it's blue and purple and also dun sa kabila, it's blue and purple. Okay? So there is random assortment already. Okay? So at anaphase 1, you would now see the division. You would now have the blue and the purple one mixed to each side. Okay? So at telophase 1, there's already a variation or it is now already different from the parent cell. Okay? So at this, the first stage or first division of your meiosis 1 is very critical. Okay? So it is responsible for the the new genetic combination. Okay? So in the first nuclear division, duplicated homologous chromosomes line up and cross over. Okay, that's the key term, crossing over. Then they move apart toward opposite spindle poles. Okay. Now at this point, two nuclear envelopes form around the clusters of still duplicated chromosomes. Okay. So yan, ito yung closer look for crossing over. So let's say yung blue is from the father, the purple is from the mother. So nag-cross over, nag-mix yung traits or alleles, and now you have new combinations of the parental alleles. Okay? Now in meiosis 2, this is the second nuclear division that separates the sister chromatids. And now, four haploid nuclei would be formed. Okay? Each one with complete set of unduplicated chromosomes. Okay? So basically, for the second division, it's like mitosis. Okay? Same lang siya ng stages. I mean, the events are the same. But basically, the your end product would be haploid daughter cells. Okay, I hope that's clear that your PMAT2 is almost the same with your mitosis, but the difference is that the chromosomes are already, or there's already genetic variation. Okay, now there is no DNA replication between the two nuclear divisions, so nag-continue lang siya to the division. Okay, further divide lang siya, okay? So, pero yung DNA hindi na siya nag-replicate. Meaning, after meiosis 1, hindi na siya pumasok ng interphase. Okay? So, after your telophase 1, diretso siya sa prophase 2. Okay? So to summarize mitosis and meiosis, here is a diagram comparing the two divisions. So for mitosis, you simply have four stages. You have the prophase, metaphase, anaphase, and telophase. And from one parent cell, you would have two daughter cells which are identical to the parent cell. Okay? Now for meiosis, you would have two divisions. You have the prophase 1, metaphase 1, anaphase 1, telophase 1, or the PMAT 1. Then you have now another round. You have the second division or the PMAT2. And as you could see, in prophase 1, there is a crossing over of their genes. And at metaphase 1, there is random assortment and the shuffling further of your chromosomes. Okay? So at anaphase 1, you would see already the distribution of the variation of your chromosomes. Okay? So at the end of meiosis, you would now have four daughter cells which are haploid and they are not identical to the parent cell. Okay? So basically, for the second division of your meiosis, it's the same with your mitosis, but again, the difference is the chromosome number. Okay? So sa mitosis, it's still the daughter cells would have diploid chromosomes, while for the telophase 2, for your meiosis, you would now have haploid cells. So that's all for cell division. So I hope you would be able to categorize or characterize now mitosis from meiosis. Okay? So we'll see you in our next video. Bye!